Immunology Insights, Basics and Beyond, Chapter 49, HIV, AIDS and the Immune System. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we honored Ralph Steinman and Harald zu Hausen, two pioneers who paved the way for cancer vaccines and immunotherapies. Today, we shift from cancer to infectious disease immunology, beginning with one of the most defining pandemics of modern history, HIV and AIDS. This chapter explores how HIV dismantles our immune defenses, why this leads to deadly opportunistic infections, how treatment transformed the outlook, and what new strategies, including lessons from naturally resistant individuals, are teaching us about immunity itself. So let's talk science. What is HIV and AIDS? HIV, the human immunodeficiency virus, is a retrovirus that infects and gradually destroys the immune system's CD4 positive T helper cells, the orchestra conductors of adaptive immunity. Without them, immune responses become uncoordinated and ineffective. If untreated, HIV progresses to AIDS, acquired immunodeficiency syndrome, marked by critically low CD4 counts and susceptibility to infections and cancers that healthy individuals can usually fight off. Since its recognition in the early 1980s, HIV has infected more than 80 million people worldwide and claimed over 40 million lives. Thanks to modern therapy, it is now a chronic, manageable disease, but still without a cure. How does HIV infect the immune system? Well, the virus is a master at hijacking immune cells. First, we have the entry. The viral GP120 protein binds to CD4 receptors with CCR5 and CXCR4 acting as co-receptors. This specificity makes CD4 positive T helper cells the primary targets, though macrophages and dendritic cells can also be infected. Second, we have fusion and integration. GP41 enables fusion. Reverse transcriptase converts viral RNA into DNA and integrase inserts it into the host genome, turning the cell into a permanent virus factory. Third, let's look at the roles of the various cells. CD4 positive T cells are destroyed or functionally impaired, collapsing immune coordination. Macrophages and dendritic cells serve as viral reservoirs and transporters, spreading HIV across tissues. CD8 positive T cells mount an initial attack but eventually become exhausted, and B cells lose proper support, leading to dysfunctional antibodies. This progressive disruption means that while some immune functions persist, the adaptive system loses its command and precision, making defense against infections and cancer unreliable. So let's have a look at these opportunistic infections and cancers then. In a healthy individual, pathogens like fungi, mycobacteria, or certain viruses are usually controlled. In AIDS, they thrive. Let's have a look at some examples of opportunistic infections first. Pneumocystis jirovecii pneumonia, which is a fungal lung infection. Tuberculosis, which is particularly aggressive in HIV patients. Cytomegalovirus retinitis, which leads to blindness. And fungal meningitis. Next, some examples for opportunistic cancers. Kaposi sarcoma, driven by HHV8, which is the human herpes virus 8. Certain lymphomas, for example EBV associated, and cervical cancer due to impaired immune surveillance of HPV. These are not random diseases. They reflect exactly where immune defenses are weakened. Lack of CD4 help cripples CD8 killing and antibody maturation, while chronic immune activation creates fertile ground for malignancy. What is the clinical course of HIV? Well, the disease typically unfolds in three stages. First, we have the acute infection, flu-like illness, very high viral load, and sharp drop of CD4 cells. Second, we have the chronic phase, or clinical latency. The virus persists at lower levels, CD4 counts slowly decline, and this phase may last years without symptoms. And third, we have AIDS. Below around 200 CD4 cells per microliter, opportunistic infections and cancers appear. Without therapy, most patients progress to AIDS within 8 to 10 years. From fatal to chronic, the treatment and control of AIDS. In the 1980s, an HIV diagnosis was almost uniformly fatal. The breakthrough came in the mid-90s with the combination antiretroviral therapy, or ART. ART works by combining drugs targeting reverse transcriptase, protease, and integrase, often with entry inhibitors. Using 3 or more drugs prevents resistance. Thereby, ART suppresses the viral load to undetectable levels, allows the CD4 cells to recover, and transforms life expectancy to near normal. Here, the U equal U principle counts. Undetectable means untransmittable. Suppressed patients do not transmit HIV sexually. Today, more than 25 approved drugs exist, and long-acting injectable therapies are emerging. But ART must be lifelong, because latent reservoirs remain untouched. How does prevention work and what are future directions? Stopping HIV requires both therapy and prevention. We have PrEP, pre-exposure prophylaxis, daily or long-acting drugs like tenofovir, emtricitabine reduce acquisition risk by over 90%. Then we have vaccines. None are licensed yet, but trials using mosaic antigens, broadly neutralizing antibodies, or BNAbs, and even mRNA vaccines are ongoing. And then we have functional cure research, like shock and kill, so wake up a latent virus and eliminate it, or block and lock, so silence HIV genes permanently. Interestingly, there are some naturally resistant individuals, and we get clues from immunity. Some rare individuals provide hope for new approaches. First, we have the CCR5 delta 32 mutation, a genetic deletion that prevents HIV entry via CCR5. People homozygous for this mutation are highly resistant to HIV infection. This insight led to CCR5 blocking drugs and inspired gene editing strategies. Second, we have elite controllers, a tiny group of HIV positive individuals who suppress the virus without therapy, likely through especially potent CD8 positive T cells and immune regulation. Studying them may reveal natural pathways to durable control. These examples show that the human immune system can defeat HIV. It's just rare. If science can reproduce these mechanisms, a cure becomes possible. Of course, a disease like this has high clinical and scientific relevance. HIV and AIDS reshaped medicine and immunology. It revealed the central role of CD4 positive T cells, exposed the devastating consequences of immune collapse, and spurred the development of antiviral therapies that are now used for many other infections. It also galvanized global health, creating the largest coordinated treatment programs in history. Equally important, HIV continues to drive innovations in vaccine, in gene editing, and in our understanding of chronic immune activation and exhaustion. In short, HIV and AIDS are not just a public health challenge, they are teachers pushing immunology forward. To conclude, from a mysterious deadly new disease in the 1980s to today's chronic but manageable condition, the progress has been enormous. Still, the virus's ability to persist and evade cure means the fight is far from over. By learning from both therapy and nature's own resistant individuals, the future may hold not just lifelong control, but perhaps even a cure. In the next chapter, we will turn to another major pathogen, tuberculosis, and explore how the immune system fights this ancient foe and why it remains one of the deadliest infections worldwide. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.